All right, so this is our second lecture for chapter 28. In lecture 1, we looked at the difference between monocots and eudicots. We looked at plant organs and the organ systems that they're contained within, and now we're going to look at the tissues that make up those organs and the cells that make up the tissues. So 3 main tissue types in plants are our dermal tissues. This is our outermost covering, and so it's blue. You'll see it as blue throughout the text. Our vascular tissue, which is purple. This transports our materials. We've talked about our xylem and phloem as far as our vascular tissues. And so all the purple delineates where you will find vascular tissue. And then finally is our ground tissue. These are our specialized cells. And so they are going to be the yellow tissue that fills in between the dermal and vascular tissues. And so each tissue continues throughout the system, as you can see. The colors will delineate each tissue as we move through. Our dermal tissue is that outer protective covering in non-woody plants. So non-woody plants, this outer dermal tissue is called the epidermis. So it is a layer of tightly packed cells that prevent things from getting in and out of the cell, help to protect the plant. In woody plants, it is called a periderm. And so protective tissues called periderm replace the epidermis in the older regions of a stem. So they have epidermis when they're young, but then it is replaced by the periderm. And that is what we constantly think of about wood. So when we think of wood, that is the periderm in our woody plants. On leaves, the epidermis is the cuticle, that waxy coating that prevents water loss and makes the plant more durable. And one of these protective features that we talked about earlier, trichomes. These are hair-like outgrowths of the shoot. They help with reduce of water loss, reflect light, and defend against herbivores and pathogens. So they are a mechanical barrier. Plus, they secrete distasteful chemicals or harmful chemicals that will keep herbivores and insects from eating the plants. Our vascular tissue, and we've talked about these a couple of times, are our xylem and our phloem. The xylem is going to conduct the water and dissolved minerals, whereas the phloem will conduct the sugars or carbohydrates made by the plant via photosynthesis. The still, this is our vascular tissue or root system. So the still is the composition of all of that vascular tissue. In angiosperms, the root still is a solid central vascular cylinder of xylem and phloem, whereas in the stems and leaves, it is usually divided into vascular bundles. So here you can see multiple bundles in our leaves and our stems, and the single cylinder of vascular tissue in our roots. But both are made up of xylem and phloem. And then last is the yellow tissue, our ground tissue, has specialized cells. These are where our metabolic functions take place. And so the pith is the ground tissue internal to the vascular tissue. So this would be the pith. And then the cortex is that external. And so this would be the cortex, or this would be the cortex. But ground tissue, that specialized tissue for photosynthesis, metabolic functions, short distance transport, storage, or support. The 3 cells that you're going to find that make up our ground tissue are our parenchyma. And so this is our metabolically active cells that are going to function in those photosynthetic and healing and carry out those metabolic functions within the cells. And here's what it looks like. You can see very thin walls, lots of space for metabolic function to take place. Parenchyma, you can see a slightly thicker cell wall, but it still provides flexible support in the growing parts of the plant. And then our third is the sclerenchyma. This is a rigid support. So this will cause rigid support. See how much thicker these cell walls are. They contain lignin, and they are a strengthening polymer. And this is what you're going to find more in our woody plants. So let's watch a quick review of a plant cell, just to remind us of the specialized feature of a plant cell.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
All right, so now let's look more closely at the cells. Again, with the parenchyma cells, you have this very thin outer cell wall. They are flexible, generally lack any secondary walls to the primary cell wall, that large central vacuole that is unique to our plant cells, and they perform most metabolic functions, retain the ability to divide and differentiate. So they can change into other types of cells. Parenchyma, they have the unevenly thick cell walls, thicker than what we see with the parenchyma, provide flexible support without restraining growth, are living at maturity. So they're still alive, still have an active nucleus. Whereas our sclerenchyma cells are those rigid, they are actually mainly cell wall. They are dead at functional maturity, and so they have sealed in and no longer function. There are two types of our sclerenchyma cells. Our sclerids are boxy, irregular in shape with thick lignified secondary walls, and fibers are long, slender, and grouped in strands. Our vascular tissue are made up of these two types of cells. For our water-conducting cells, and water is carried in xylem, they are dead lignified, so lignified just like we saw with our sclerenchyma, and sclerenchyma cells. So lignified just like our sclerenchyma cells. And there are two types of water-conducting cells. The tracheids, these are the long, thin cells with tapered ends that move water through pits. And so these little openings in the wall, so they move from one tracheid to the next through these adjoining pits. And then your vessel elements, so here are our vessel elements, align end to end. So rather than having these tapered ends that slide into one another, these guys join end to end like long pipes called vessels. So our vessel elements common in most angiosperms and a few gymnosperms, and then a few seedless vascular plants. And the tracheids occur in the xylem of all vascular plants. So tracheids is in everybody, vessels mainly in angiosperms, but some gymnosperms and a few of our seedless Seedless. Remember, those are our ferns and stuff. Seedless vascular plants. And then our sugar conducting cells. These are going to be our sieve tube elements. So in angiosperm, sugars are transported in the sieve tubes. These are chains called sieve tube elements, sieve plates. These guys right here are what divide the sieve tubes and allow fluid to flow between cells. The sieve tubes also have companion cells. These are the guys on the outside with the purple nuclei, nucleus of the companion cell. The sieve elements themselves lack organelles, but they have these companion cells whose nucleus and ribosome serve both cells. So keep everybody alive and happy via diffusion. And here you can actually see a photomicrograph of those cells. All right. So we've gone over all of the tissues and cells. How do these grow? What is the purpose of the cells? And so we have some plant organs, like animals, only grow to a certain size. And so that is determinate growth. So you have a self-determined size. The rest of the plant continues to grow throughout its entire life. And this is unique to plants, and you don't see it in animals. And this is indeterminate growth. So this means that you continue to grow until you die. And where you have this indeterminate growth is called our meristems. And so tissues that grow to elongate the plant during favorable conditions. And we have our apical meristems. Remember, we saw this back on another diagram. And so it grows from the tip. The cell divides, daughter cell in the primary. And then they differentiate into our different elements. So these guys, the brown, are undifferentiated. They are continuously dividing, and as they divide, they will push down, or actually it keeps pushing forward, and the cells that had just divided, one of the daughter cells, so one goes to become one of the other cell types, whether it be our ground tissue, our dermal tissue, or our vascular tissue. And see, we still have this color coding. Dermal's the blue, ground's the yellow, and our conducting cells, or our vascular cells, are the purple. And so over time, you can see how it grows. And so it's always pushing through the soil like a root cap to help protect the apical meristem so it can push through the soil and through rocks and through other things. Don't have to have a cap on our stems because the stems are usually just pushing through air. And so there are two main types, and we looked at these earlier. We have the apical, and remember the apical is going to be located at the tips, api meaning tip. And then you can also have, and so these apical meristems are going to keep pushing and elongating shoots and roots in a process called primary growth. And so length is called primary growth, done at the apical meristems that are found at the tips of the roots and the shoots. And you can see all the little brown spots on the end of the roots. We have these axillary that come out at the nodes, and then the apical at the tip of the shoot. So all these little brown areas are where we have this primary growth in our apical meristems. And then we have the lateral meristem. And so lateral is going to create thickness, apical length, lateral thickness. This is called secondary growth. So lateral is going to be secondary growth that increases width. Apical meristems are going to create length, and it is primary growth. We have two types of lateral meristems. These are the ones that are going to increase the width. We have the vascular cambium. Vascular cambium adds layers of vascular tissue called secondary xylem. And so vascular cambium is going to add secondary xylem, which is what we call wood, and secondary phloem, which we usually call bark. And so that secondary phloem, the cork cambium, replaces the epidermis with that periderm, which is the thicker and tougher type of dermal tissue. So we're going to increase in circumference. And here you can see we have our pith. Remember, pith is any of the ground tissue inside the vascular tissue. And here the red and the blue represent our vascular tissue. Or the red is going to be the vascular. The blue is going to be our dermis. And in between the vascular and the dermis is the cortex. And so each of these are going to divide. The vascular tissue will divide and add cells in both directions. It adds secondary xylem towards the pith, and it adds secondary phloem towards the cortex, or into the cortex. And then we can see also that our cork cambium, which is our dermal tissue, is going to be adding on cells, which will become the periderm. And so most of the cells are secondary xylem. So you can see how many more are the secondary xylem, are wood, and the cells in the cortex stretch and are eventually crushed, becoming more compacted. All right, so this is our secondary growth in our lateral meristems, and then recall back that primary meristem, the brown tips that we saw, as that goes and the one daughter cell goes down into growing the plant itself, it differentiates into one of the three tissues. So we have three primary meristems. Each of those can become a protoderm, which produces dermal tissue. Ground meristem produces ground tissue, and that procambrium, just like we see out here, the cambrium, produces vascular tissue.
Some plants get so tall that they would topple over if they did not also get wider. Secondary growth enables these plants to increase their girth significantly to support their added height and weight. Widening occurs at lateral meristems, also called secondary meristems or cambia. We'll use a longitudinal section and a cross-section of a stem to illustrate the process of secondary growth. In this view, you can see a ring of vascular bundles between the pith and the cortex. The epidermis is the outermost layer of the stem. When you look at a detail of an individual vascular bundle, you can see that there is a layer of cells between the primary xylem and the primary phloem. This layer consists of undifferentiated cells. It is a lateral meristem known as the vascular cambium. During the growing season, the cells of the vascular cambium divide, forming rows of cells on the inside that mature into secondary xylem. The cambium cells also divide to form rows of cells on the outside, where they mature into secondary phloem. These secondary vascular tissues add both conductive capacity and support to the growing stem. The vascular cambium produces secondary xylem and secondary phloem during each growing season. As the vascular cambium cells divide, the outermost stem tissues must expand to compensate for the increased internal girth and to retain continuity of the stem's circumference. Another lateral meristem known as the cork cambium develops in the outer layers of the cortex. The cork cambium divides in a manner similar to the vascular cambium and produces layers of protective cork cells on the outside. This plant has now completed 2 years of growth. As the vascular cambium produces new secondary xylem and secondary phloem cells, the circumference of the stem increases. The epidermis, cortex, and primary phloem are pushed outward, crushed, and eventually sloughed from the stem. The new cork cambium produces cork cells, while the vascular cambium continues to generate new secondary xylem and phloem.
All right. So let's finish up Lecture 2 with going back over our apical meristems. So we have those 3 primary meristems, the protoderm, the procambrium and the ground meristem. So that brown cell at the tip differentiates into one of these three. The protoderm will go on to form the epidermis. The procambrium will be the primary phloem and the primary xylem. And then the ground meristem will go into the ground tissue or the pith cortex. So ground tissue made up of the pith inside the vascular tissue, cortex outside the vascular tissue, between the vascular tissue and the dermis. All right, so then we continue growth from the primary tissues into what we call lateral meristems. So the procambrium in the secondary growth, the procambrium can form the vascular cambrium. The cortex is going to form the cork cambrium. And then the cork cambrium can form cork or the periderm, or the cork cambrium can make the cork, and the two together make the periderm. And the vascular cambrium is going to make that secondary phloem, pushing out towards the dermal tissue, and the secondary xylem, which is laid down towards the pith in the pith area, making up our wood. So the secondary xylem becomes what we know as wood. And those rings, each of that ring that it was showing, that's when we count how many years old it is. Each ring represents a year's worth of growth. And so there is a breakdown of the apical meristem, our primary growth, our primary meristems and tissues, and then our secondary growth from our lateral meristems and those secondary tissues.